Hello guys, this is going to be a little bit different. I'm sorry I haven't been able to be there with you, but I'm going to try and do something a little revolutionary here. We're going to try and do this through video and the magic of YouTube. So we are going to work on a few notes today. You guys I know have done a ton of things using photosynthesis and cellular respiration. So today I'm going to try and help you guys out. We're going to kind of work through some of these things together. I know you can't answer or ask me any questions today, but hopefully, if you guys still have questions at the end of all this, I will be back in school on Monday and we can kind of go over whatever we need to do. So today, sit back, enjoy the fun notes. You're going to need some colored pencils. We'll get those out for you in a little bit and we'll see how this actually works. All right, guys. First thing you need to do is get out the packet. There is actually a handout that's called Chapter 9, Energy in a Cell. And we're going to kind of work on this a little bit together and see what you guys come up with. All right, guys, look for it right now. Chapter 9, Energy in a Cell. So what you guys now see in front of you is a concept map that's going to use a bunch of terms that you guys have been learning about with photosynthesis, and we're going to try and use those terms to fill in the 12 blanks that you guys see in front of you. Now it takes a little bit of time. It's not the easiest thing to do. So I'm going to kind of start you off, get you guys thinking a little bit about this. One of the words, oxygen, you'll see at the very top of the page was actually incorrect. So I had to replace it with the word energy. It actually makes it work a lot easier if you see that word up there. So here's what I'm going to do. Let's do the first one together, and then you guys can try and work on this for a couple minutes by yourself or with the person sitting next to you, and then we'll kind of come back and we'll kind of go over it. So one of the things you learned about in photosynthesis is that there are these things called thylakoid membranes. They are parts of chloroplasts. So for number one, thylakoid membranes are found in chloroplasts. So we're going to put that right in here. Well, that didn't work very well, did it? But we're going to stick with it for now. So thylakoid membranes are found in chloroplasts, and another thing that is found in chloroplasts are these things called stroma. So now that you guys have those two, take a couple minutes, pause the video, and see if you guys can now finish the rest of the information on here. It's not the easiest thing, but I think that you guys, now that you've seen a couple of these PowerPoints, we talked about it a little bit at the beginning of the week, should be able to fill it in. Go ahead and see what you guys are able to come up with, and then hit pause or play when you guys feel like you're ready to go on. Alright, now that you guys have kind of worked through this a little bit, let's see if you guys came up with the same answers that I was able to come up with sitting here in my beautiful living room staring out 3 to 5 feet of snow, depending on where you're looking. So here's the first word. Thylakoid membranes. They contain chlorophyll. So we'll put on there chlorophyll. So chlorophyll obviously is what traps Earth's or the sun's sunlight. And it's going to provide the energy for our light-dependent reaction. Which converts light energy into a form of chemical energy. And you guys know that's in the form of ATP, that special molecule that we keep talking about over and over and over again. So if we go back up to the top, chlorophyll is going to pick up the electrons from the water that it brought up through its roots, the capillary action, and the reactants for photosynthesis, as you guys know, are water plus carbon dioxide. Excellent. That water is going to get split into oxygen and a hydrogen ion. So there's actually oxygen is used, but you can see it's actually already at the top. So that's why they just put it on there twice, which is the mistake they made. So then at the very end, the Calvin cycle, this whole process that we're talking about, is used to produce glucose. That monosaccharide that we talked about a long time ago when we were doing things with biochemistry. So hopefully now this page kind of makes sense to you guys, and you were able to fill it in based upon what you've been seeing and learning about over the past couple classes. So that's the first page that we're going to look at. Now if you flip it over, we're going to practice using some of our graphing skills and help you guys to use some of the different things you now know about photosynthesis to answer those questions. Now that you guys have flipped the page, take a second to read the short little section at the top that talks about two factors affecting photosynthesis. Make sure that when you guys read them, that if you're underlining any of the big vocab terms that we've been talking about over the past couple classes. So go ahead right now, take about 30 seconds on your mark, get set, go. All right, that's a little less than 30 seconds, but good enough for us today. So, here's what we're going to look at. Question number 1. What does the graph on the left tell us about the effect of light intensity on the rate of photosynthesis? So if I were to look at this graph, I can see that it starts and it starts to move up. So I kind of follow it along. So if I were to look at this, there's a couple different things that it's going to tell me, but one of the most important things it tells me is that when I get to about this point here, you should notice something about the rate of photosynthesis. It doesn't really increase at all at that point. So after about 9,000 lumens, not a lot is changing. So what I'm going to write down here for myself is that at 9,000 lumens, and if you remember from the reading up above, lumens is just a way of measuring light intensity. So at 9,000 lumens, the rate of photosynthesis stays the same. Perfect. So what happens when the light intensity rises above 9,000 lumens? Well, we already kind of wrote it above. It makes it pretty easy. We just have to put in there that it stays the same. Excellent. So now let's look at questions number 3 and 4. So question number 3 asks you guys to think about this. What adaptive advantages would a plant have if its photosynthetic rate kept increasing with light intensity above 9,000 lumens? So if I'm a plant, obviously what I want to do is I want to make as much food as possible with the available sunlight that's given to me. So if I'm a plant that has the ability to keep increasing the amount of energy I can create after 9,000 lumens, that's going to be a great thing for me because I can make more food, I can store more food, and if there's a drought or something changes in the environment, I have an advantage that other organisms may not. So what I'm going to write on here is that the plant could make more food. Excellent. Now let's look at the other graph. What does the graph on the right tell about the effect of temperature on the rate of photosynthesis? So when I look at this graph, I can see a bunch of different things on here. So I can see that there's obviously temperature down here on my x-axis. I've got the rate of photosynthesis over here on my y. And I notice that as the temperature starts to increase, I get to a certain point where all of a sudden around 30 some degrees, 33, there's this rapid crash and my rate of photosynthesis comes screeching to a halt. So if I look at over here, what does the graph on the right tell me about the effect of temperature on the rate of photosynthesis? Basically, that it increases until you hit 33 degrees. Excellent. That's 33 degrees Celsius. So what happens when the temperature rises above 33 degrees Celsius? Well, obviously, when we look at our chart, it's pretty simple. Photosynthesis decreases. So that's all I really need to write over here. Photosynthesis decreases. Excellent. All right, now let's go look at the next couple pictures that you guys have in front of you. As you and I just kind of noticed, we understand that as temperature increases, there gets to be a certain point around 33 degrees, where there's this rapid decrease then in the ability of a plant to go through photosynthesis. So the question they're asking us is what might cause this change? Obviously, there's lots of things that could cause it, but I want you to think of plants kind of like people. And people have to maintain a stable internal environment. And what do you guys call that? Somebody in the room should have said homeostasis. Excellent. So just like in people, plants have to go through homeostasis as well. They have to maintain that stable internal environment. So for us, we maintain a stable internal environment of about 98.6 degrees Celsius. Well, for plants, they have a homeostasis, at least the things that work best for them, when they get to around 33 degrees. So at this temperature, their enzymes are working the best, everything is kind of working the way it's supposed to, and those plants are able to best use the sunlight that they're getting as well as the soil nutrients and the water and all that kind of fun stuff in the atmosphere like carbon dioxide. So what might cause the change to decrease there? Well, what we're going to say is that the enzymes begin to break down. Another term that sometimes you will hear people use is to say that they are denatured. And that's the reason why parents and people are really concerned when you guys get a fever above 100 or some degrees, because the same things happen to your enzymes. They start to break down and not work as well. This is kind of like getting a fever for a plant. All of a sudden, the temperature gets to a certain point where they're no longer able to do the things that they need to do, and photosynthesis begins to stop. Now, let's look at the very last question on here, number 7. What light intensity and temperature levels allow the highest photosynthesis rate or photosynthetic rate? Well, we've already kind of circled one. So if I look up here, it's about 9,000 lumens. And the temperature, as we looked at over here, was about 33 degrees. So it makes it really simple. So it would be for light, we know that it's going to be about 9,000 lumens. And then for temperature, it's going to be about 33 degrees Celsius. Excellent. So now we know a little bit more about how enzymes help out with photosynthesis and helps to maintain homeostasis in a plant. Now we are going to kind of change gears and we're going to get the colored pencils out. So everyone's going to need probably about 7 or 8 different colored pencils. You can get them from the back of the room, or you can go grab your own real fast, but probably just use the ones in the back of the room. It'll be easier for everybody involved. All right, guys, get your pencils and then head on back and I'll show you what we're doing next.